Every cell in your body carries a countdown, and each time it divides, the protective caps on its chromosomes get shorter until the cell stops repairing and starts dying. Right now, a plant growing across 3 continents holds the compound that rebuilds those caps back. And at the end of this video, you will know which 2 plants reset your cells and why no supplement company will ever say their names. Your body replaces roughly 330 billion cells every single day. Each new cell copies the old one, but the telomeres at the end of every chromosome lose a small piece with every division. Think of them like the plastic tips on a shoelace. When the tip wears down, the lace unravels. When the telomere gets too short, the cell can no longer divide. It enters a state scientists call senescence. It stops working. It starts releasing inflammation into the surrounding tissue, and it never recovers. This is not disease. This is aging measured at the molecular level. But there is a second collapse happening at the same time. Your body maintains a reserve of stem cells and progenitor cells whose only job is to repair damaged tissue and replace what is lost. These cells also age. They slow down. They lose the ability to regenerate bone, brain tissue, muscle, and skin. The wounds that healed in days when you were 20 take weeks when you reach 60. That is stem cell exhaustion, and it accelerates every year after 40. These 2 processes are not separate problems. Telomere shortening and stem cell decline are the same biological failure measured on 2 different scales. Fix one without the other, and the cell still collapses. Address both, and the entire repair system has a chance to function the way it did decades ago. The pharmaceutical industry understood this before you did. In 2011, a company called TA Sciences began selling a telomere supplement called T65 for up to $600 per bottle. The active compound inside that capsule was extracted from a single plant. That plant grows wild in the grasslands of northern China and Mongolia. You can buy a pound of its dried root for less than $25 at any traditional Chinese herbal shop in any Chinatown on Earth. The markup is not a business model. It is an information gap, and today that gap closes. The plant is called Astragalus membranaceus. In Chinese medicine, it has been used for over 2,000 years under the name Huang Qi. It is a perennial legume with pale yellow roots that look like dried sticks of driftwood pulled from a riverbed. It grows in sandy soil across the steppes of Inner Mongolia and the highlands of Shanxi province. It fixes nitrogen in the ground the same way clover does, meaning the earth it grows in becomes richer every season. And inside that unremarkable root sits the compound that launched an entire anti-aging industry. The compound is called cyclotragenol. In 2011, Calvin Harley and his research team published a study in the journal Rejuvenation Research demonstrating that TA65, a purified extract of cycloastragenol from astragalus root, activated telomerase in human immune cells. Telomerase is the enzyme your body uses to rebuild telomeres. It is the only known mechanism that adds length back onto the chromosome cap after cell division removes it. Harley found that treatment with this extract increased telomere length in the shortest and most vulnerable telomeres without elevating cancer risk markers. That same year, Bruno Bernardes de Jesus and a team at the Spanish National Cancer Research Center in Madrid confirmed the results in aging mice. TA65 rescued critically short telomeres across multiple organ systems, reduced DNA damage signaling, and improved both insulin sensitivity and bone mineral density. The animals did not simply live longer, they aged slower at the cellular level. Their tissues behaved younger than their calendar age. Then came the human trial. In 2016, Laura Salvador and colleagues published a one-year, randomized, double-blind, placebo-controlled study in the journal Aging. 117 human participants between 53 and 87 years old received TA-65 daily for 12 months. At the end of the trial, the treatment group showed a statistically significant decrease in the percentage of critically short telomeres in their white blood cells. This was the first published clinical evidence that a plant-derived compound could measurably lengthen telomeres in living humans. And the benefits extend beyond the telomere itself. Separate research published in the Journal of Ethnopharmacology by Lee and colleagues in 2014 demonstrated that astragaloside-4, another active compound found in the same root, suppresses the inflammatory signaling pathway known as NF-kappa B. Chronic low-grade inflammation is one of the primary accelerators of telomere erosion. By calming that inflammation, astragalus does not just rebuild the protective cap, it slows the very force that grinds the cap down. The root is traditionally harvested after 4 to 5 years of growth. Herbalists across Beijing and Shanghai slice it thin and simmer it for 20 to 30 minutes into a pale golden broth that tastes faintly sweet. You can buy the dried slices at any Chinese herbal pharmacy or order them online for a fraction of what the supplement costs. You can also grow it yourself in a deep container with at least 12 inches of well-drained sandy soil. It survives frost. It returns every spring stronger than the year before. And because it is a nitrogen-fixing legume, the soil around it improves with each season. But telomere protection is only half of the equation. A cell with a long telomere still needs to divide to repair tissue. And cell division at the repair level depends on the stem cells and progenitor cells that drive regeneration. The next plant does not protect your cells. It wakes them up. You may have already eaten this plant without knowing what it does inside your body. If you have ever walked into a Korean barbecue restaurant in Seoul, or Los Angeles, or London, you have seen the broad green or purple leaves served in a stack alongside the grilled meat. You wrap the pork or beef inside the leaf and eat it whole. In Korea, they call it dolkai. In Japan, it is shiso. In Chinese medicine, it is zisu. Across East Asia, it is as common as basil is in Italy. In the West, most people have never heard its name. The plant is Perilla frutescens. It is an annual herb in the mint family, and it contains 3 compounds that peer-reviewed research has linked directly to the proliferation and survival of stem cells in aging tissue. The first is rosmarinic acid. In 2016, Fonteles and colleagues published a study in Neurochemistry International showing that rosmarinic acid enhanced neurogenesis in the hippocampus of adult mice. Neurogenesis is the formation of new functioning brain cells from neural stem cells. This process declines sharply after middle age and is directly connected to memory loss and cognitive deterioration. The rosmarinic acid concentrated in perilla leaves reactivated that process in adult brains that had already begun to slow. The second compound is luteolin. In 2013, Zhu and colleagues published in PLOS1 that luteolin promotes the differentiation of neural stem cells into mature functioning neurons while simultaneously shielding them from inflammatory damage. 6 years later, in 2019, Kai and a team from Nanjing Medical University demonstrated in the Journal of Neuroinflammation that luteolin suppresses microglial activation. Microglia are the immune cells of the brain. When they become chronically overactive, they destroy the very stem cells that would otherwise repair aging neural tissue. Luteolin calms them down and gives the stem cells room to work. The third compound is apigenin. Kang and colleagues published in the International Journal of Molecular Sciences in 2017 that apigenin promotes osteogenic differentiation of mesenchymal stem cells. In plain terms, it directs bone marrow stem cells to build new bone tissue. This matters because osteoporosis is not a calcium problem. It is a stem cell signaling failure. The raw material for bone renewal exists inside the marrow. The signal telling stem cells to activate weakens with age. Apigenin restores that signal. And then there is a fourth dimension. Perilla seed oil contains between 54 and 64% alpha-linolenic acid. That is one of the highest omega-3 concentrations of any plant on Earth. Kim and colleagues published in the Journal of Medicinal Food in 2014 that perilla oil supplementation reduced inflammatory cytokines and boosted antioxidant enzyme activity in human subjects. This matters because stem cells cannot proliferate in chronically inflamed tissue. The inflammation shuts them down before they can do their job. Perilla reduces that inflammation while simultaneously feeding the regenerative machinery. Growing perilla requires almost no effort and almost no space. It germinates from seed in a small pot on a sunny windowsill. It tolerates partial shade. It self-seeds so aggressively that a single planting produces harvests for years without replanting. The leaves are eaten raw in wraps, pickled in soy and sesame, or brewed into a light herbal tea. The seeds are pressed into oil or ground into seasoning powder. Every part of the plant is edible. You can find fresh perilla at any Asian grocery store in any major city from New York to Melbourne. 2 plants. Astragalus protects the telomere so the cell can keep dividing. Perilla supports the stem cells that perform the division. One guards the clock, the other winds it back up. But these 2 are not alone. 5 more plants are growing right now, each one targeting a different axis of the same cellular collapse. Each one is backed by published research and is available to anyone willing to grow it.
Walk into any health store in America today and you will see an entire wall of supplements claiming to fight these forces. Fisetin capsules, resveratrol pills, telomerase activators, autophagy boosters, each one costs between $40 and $200 a month. But here is what the companies selling those bottles hope you never realize. Every single compound on that shelf was first found inside a living plant. They extracted it, they concentrated it, they packaged it with a label and a barcode, and they priced the average person out of the solution. The source plants still grow in backyards and roadsides and ditches across the world. They cost pennies. They grow themselves back every season, and nobody selling you a bottle has any reason to mention that. The first plant in this system is something you have almost certainly eaten before, but you ate it wrong. You bought it fully grown at the grocery store, you steamed it on a dinner plate, and you were getting some benefit, a little. But the compound that activates over 200 protective genes in your body is concentrated almost entirely in the sprout. The tiny sprout that is 3 to 5 days old contains up to 100 times more of this compound than the fully grown vegetable sitting in your refrigerator right now. The compound is called sulforaphane. It is the most powerful known natural activator of your body's master defense switch. It also activates autophagy. It is the exact recycling system that Osumi won the Nobel Prize for discovering. The plant is broccoli and the form that matters is the sprout. Grow them in a mason jar on your kitchen counter. Soak a tablespoon of broccoli seeds overnight, then drain them. Rinse them twice a day. In 3 to 5 days, you will have a full jar of fresh sprouts ready to eat. No soil, no sunlight, no garden required. Eat them raw or blend them into a smoothie. Keep 3 jars in rotation and you will never run out. Broccoli sprouts activate your body's recycling system. But there is a fruit you have almost certainly eaten this month, and the part you throw away contains the most powerful compound ever tested for destroying zombie cells. When the Mayo Clinic tested 10 different flavonoids head-to-head, one destroyed zombie cells more effectively than all the others. That compound is called phycetin, and the richest natural source of phycetin on Earth is not some exotic berry from a distant jungle. It is the strawberry. Mice given phycetin late in life lived roughly 10% longer and showed fewer age-related problems than the control group. That result did not come from a supplement label. It came from one of the most respected medical institutions on the planet. And the best natural source of that compound is sitting in any produce aisle right now for a few dollars a basket. But here is what nobody mentions. The leaf of the strawberry plant also contains bioactive flavonoids. For centuries across Europe, families brewed strawberry leaf tea as a daily tonic. Grandmothers in rural England and France kept dried strawberry leaves in their kitchens the way most people today keep chamomile. That tradition faded when companies figured out how to isolate phycetin and sell it in capsule form for $40 to $60 a bottle. The whole plant grows in a hanging basket on your porch. Alpine and everbearing varieties thrive in containers with almost no effort. Full sun, basic watering, they produce fruit continuously through the growing season and survive winter with simple mulching. Eat the berries fresh every day. Dry the leaves for tea. Freeze the extras for winter. One hanging basket replaces a supplement bottle that costs more per month than your water bill. Strawberry gives your body the compound that eliminates zombie cells. But what about the countdown clock on your chromosomes? There is a leaf brewed across East Asia for over 1,000 years that protects the very ends of your telomeres from unraveling. In Okinawa, Japan, one of the longest-lived populations on Earth, grandmothers brew this leaf every single morning before the sun rises. In the mountains of Fujian province in China, farmers have cultivated it along misty hillsides for over 1,000 years. Across Korea, it appears in Buddhist temple ceremonies that date back centuries. The people who live the longest on this planet drink it every day of their lives. The rest of the world buys the active compound in capsule form and pays 10 times the price. The plant is Camellia sinensis. You know it as green tea. The compound that matters is called EGCG. It supports telomere length and activates the very enzyme that the Nobel Prize in 2009 was awarded for. EGCG also triggers autophagy through a completely different route than sulforaphane. That means green tea and broccoli sprouts activate the same recycling system by opening two separate doors. Two plants, two keys, same lock. Brew loose leaf tea, not bagged. Loose leaf delivers significantly more active compound per cup. 2 to 3 cups a day matches the intake of the populations that outlive everyone else on Earth. For maximum benefit with less caffeine, cold brew the leaves overnight in your refrigerator. Camellia sinensis grows beautifully in a container. It prefers acidic soil, consistent moisture, and partial shade. It handles a wide range of climates, including surprisingly cold winters. Harvest the youngest leaves at the tip of each branch and dry them yourself. One tea plant in a pot produces enough for daily cups and can live for decades. Green tea shields your telomeres, but imagine a plant that goes further. One that actually reactivates the enzyme that rebuilds them. A plant so aggressive it is banned in parts of Europe. And the compound inside it makes every resveratrol supplement on the shelf look like a rounding error. This is the plant that governments spend millions trying to eradicate. In the United Kingdom alone, it can knock 10% off a property value overnight. Across Europe, it is classified as one of the most dangerous invasive species on the continent. Homeowners in London and Amsterdam panic when they see it cracking through concrete driveways and foundation walls. Entire removal companies exist solely to tear it out of the ground. And the supplement industry quietly extracts the most expensive anti-aging compound in the world from its roots. The plant is Japanese knotweed. Its root contains 50 to 100 times more resveratrol than a glass of red wine. Most resveratrol supplements already on the market are derived from this plant because no other natural source comes close. The active form is trans-resveratrol, the most bioavailable version of the compound. Resveratrol activates a longevity protein called SIRT1. This is the same switch your body flips during fasting and caloric restriction. Japanese knotweed gives you the fasting benefit without the fast. In Japan, families have eaten the young spring shoots for generations. They harvest them when they are under 8 inches tall and prepare them the way you would prepare asparagus, steamed, sautéed, or pickled in rice vinegar. The root can be dried and brewed as tea. One critical rule: never plant this in open ground, ever. Grow it in a large, sealed container only. This plant will spread through concrete if you give it the chance. In a pot, that aggression works in your favor. You get a virtually indestructible source of resveratrol that requires zero help from you. The difficulty is not growing it, the difficulty is stopping it from taking over everything else. Japanese knotweed activates the same longevity switch as fasting. But there is a golden root used across India and China for thousands of years that does something none of these other plants can do. It turns on the enzyme that actually rebuilds your telomeres. And a company figured out how to patent that compound and charge more than $100 a month for it. This is the one plant on the list you have probably already heard of: turmeric. It has been in health food stores and golden lattes across Los Angeles and New York for years now. But what most people do not realize is that the way they consume it delivers almost none of the active compound to their cells. The compound is curcumin. It selectively targets zombie cells and pushes them toward destruction. It also suppresses the master switch for chronic inflammation in your body. And chronic inflammation accelerates every single one of the 5 aging forces at the same time. More zombie cells, shorter telomeres, slower recycling, weaker mitochondria, faster sugar damage. Curcumin dials down the environment that makes everything worse simultaneously. Here is what grandmothers across India have always understood: preparation is everything. Curcumin on its own passes through your digestive system almost untouched. Combine it with black pepper and a fat like coconut oil, and absorption jumps by up to 2,000%. This is not a recent discovery. This is how kitchens from Kerala to Kashmir have prepared turmeric for thousands of years. The bioavailability problem was solved long before science had a name for it. Grow turmeric from a rhizome, not from seed. Plant it in a large container in spring. It needs warmth, moisture, and partial shade. Harvest in fall when the leaves turn yellow. Store rhizomes over winter and replant next spring. One rhizome produces a full harvest in a single season. Turmeric clears zombie cells and calms the inflammation behind all 5 forces. But what about your mitochondria? There is a fruit so ancient it appears in Greek mythology, and a clinical trial proved its compound renews mitochondria in living humans, not in mice, in actual people. In Greek myth, Persephone ate pomegranate seeds in the underworld, and it bound her to a cycle of death and renewal forever. That story is 4,000 years old. The biology behind it turns out to be almost literal. When you eat pomegranate, your gut bacteria convert compounds in the fruit into a molecule called urolithin A. This molecule triggers a process that clears out old damaged mitochondria and replaces them with fresh ones. In a clinical trial published in 2019, daily intake of urolithin A improved markers of mitochondrial health in elderly adults. A follow-up study found that middle-aged adults who took it daily gained 12% more muscle strength after just 4 months. This was the first compound ever clinically shown to improve muscle function by renewing mitochondria in living people. A Swiss biotech company called Amazentis isolated the molecule and patented it under the brand name MitoPure. Nestle Health Science bought the global distribution rights. 1 month of capsules cost between $40 and $70. The precursor compound comes from a fruit tree you can grow on your patio for the cost of a sapling. Dwarf pomegranate varieties thrive in large containers. They prefer full sun, and they are drought-tolerant once established. They are self-pollinating, so you only need one tree. Bring the plant indoors during cold winters. It begins fruiting within 2 to 3 years. Eat the fresh arils daily or juice them. Consistency matters here because your gut bacteria need regular feeding to build up the population that handles the conversion. Feed them the raw material and they do the rest. Pomegranate renews your mitochondria from the inside, but there is one more plant connected to the other Nobel Prize. It is a root used in Chinese medicine for over 2,000 years. Scientists extracted a single compound from it that reactivates the enzyme your body largely stops producing after childhood. One company patented that compound and now charges over $100 a month. In the mountains of northern China, this root has been medicine for over 2,000 years. Traditional practitioners call it huang qi. They simmer it in clay pots with whole chicken, ginger, and jujube dates. They give it to the elderly to restore what time has quietly taken away. When scientists tested the root, they found a compound called cycloastragenol that activates telomerase. That enzyme was at the center of the 2009 Nobel Prize. It is the enzyme that rebuilds the protective caps on your chromosomes. A supplement company extracted the compound, branded it TA65, and started selling it in 2008. 1 year of use was shown to increase telomerase activity in immune cells and lower inflammatory markers in the people who took it. The supplement runs over $100 per month. The source plant is Astragalus membranaceus. It grows in a deep pot in your garden for the cost of a seed packet. Start from a seed or a root cutting. Give it a deep container because the root grows straight down. Place it in full sun to partial shade. It is cold hardy. It is a perennial that returns every single year without replanting. Harvest roots after 3 to 4 years while leaving the crown intact so the plant keeps producing. Slice the dried root thin and simmer in water for 30 minutes. Add it to bone broth. Add it to winter soups. This is the same preparation Chinese herbalists have passed down for 2,000 years. Astragalus rebuilds the caps on your chromosomes, but there is another force that none of these plants have fully addressed yet: sugar. Every time you eat it, glucose permanently bonds to proteins inside your cells and stiffens them like cement. There is one flower, bright red and brewed across 3 continents, that fights this reaction harder than any plant ever tested. Before we reach that flower, there's a quiet protector that deserves your attention. When you hear about the Mediterranean diet, you think of olive oil. Olive oil gets the headlines and the shelf space, but the leaf of the olive tree contains far higher concentrations of the compound that actually matters. And almost nobody outside of traditional villages along the coasts of Crete and southern Italy pays any attention to it. The compound is called oleuropein. In cell studies, it activates autophagy, delays the onset of cellular aging, and extends cell lifespan by approximately 15%. It also supports the same sirtuine longevity protein that resveratrol targets, and it protects mitochondrial function in aging muscle tissue. Walk through a hillside village above the Amalfi Coast, and you can still find elderly women who steep olive leaves into tea every afternoon. They have done it their entire lives. They do not know the word oleuropein. They just know the tea keeps them going. Traditional Mediterranean families did not only cook with olive oil, they brewed the leaves as well. That practice faded when oil became the commercial product, and the most powerful part of the tree was left on the branch. Dwarf olive varieties grow well in large containers, and they do best in full sun. They are drought tolerant and slow to establish, but nearly impossible to kill once rooted. You can harvest leaves year-round without harming the tree. Dry the leaves and brew them as a daily tea. One small tree in a pot gives you a steady supply of one of the most studied longevity compounds in Mediterranean tradition. The tree will outlive you. Now to that red flower. In Mexico City, street vendors sell it ice cold as agua de Jamaica. In Cairo, it is carcadi poured from brass pots. Across West Africa, from Senegal to Nigeria, it is bissap. In Trinidad and Jamaica, they call it sorrel and serve it at Christmas. Same plant everywhere, Hibiscus sabdarifa. Hundreds of millions of people across the tropics brew it as daily tea. It is one of the most consumed herbal beverages on the planet, and almost nobody in the Western longevity world talks about it. Those deep red anthocyanins that give hibiscus its color are among the most powerful plant-based glycation fighters ever studied. They intercept the reactive molecules that form when sugar bonds to protein inside your cells. They trap those molecules before they can create the permanent crosslinks that stiffen tissue, damage organs, and accelerate aging from the inside out. Brew the dried calluses as iced tea or hot tea. The deeper the red, the more active compound in your cup. Daily consumption is the tradition across every culture that drinks it. This is not something you cycle on and off. This is a daily tea that civilizations across 3 continents have relied on for centuries. Grow roselle from seed in a large pot, full sun. It is a warm season annual in temperate climates and a perennial in tropical zones. Harvest the calaces after the flowers drop. Dry them and store in your pantry for year-round brewing. The seeds cost next to nothing, and one plant produces enough for months of tea. Hibiscus fights the sugar damage that stiffens your cells from the inside. But there is one final mechanism none of the other 9 plants fully cover: a compound that activates the Nobel Prize-winning recycling pathway through a completely different door than broccoli sprouts. Centenarians have unusually high levels of it in their blood, and you can sprout its richest source on your kitchen counter in 3 days. The compound is called spermidine. It was named decades ago for the tissue where it was first discovered, and the name has unfortunately kept most people from ever paying attention. That is a mistake. In studies across multiple species, spermidine extended lifespan. In large human studies, higher dietary intake was consistently linked with lower overall mortality. And here's the detail that stops researchers in their tracks. Centenarians, people who live past 100, maintain blood levels of spermidine that look remarkably similar to people decades younger than them. Their levels simply do not decline the way they do in normal aging. Something about this compound appears to keep the cellular maintenance system running long after it usually shuts down. Spermidine works by activating autophagy, the same recycling system that broccoli sprouts trigger, but through a completely different biochemical route. Consuming both means you are firing up your body's cleanup system through 2 separate pathways at the same time. 2 keys and 2 different locks opening the same door. The single richest dietary source of spermidine is wheat germ. Buy a bag for a few dollars at any grocery store and sprinkle it on food every day. But for even more benefit, sprout wheat berries or oat groats at home. The sprouting process concentrates the compound. Soak the grains overnight. Rinse twice a day. Harvest at 3 to 5 days. This is the exact same mason jar method you already use for broccoli sprouts. No soil, no garden, no outdoor space needed, just a jar on your counter next to the broccoli sprouts you are already growing. 2 jars, 2 plants, 2 autophagy pathways. Total cost is a bag of grain. Now let me show you how all 10 come together. You do not need a farm. You do not need raised beds. You do not even need a backyard. You need 1 large container, a few satellite pots, and 2 mason jars. Take a half whiskey barrel or a 25-gallon fabric pot. Set a dwarf pomegranate in the center as the tallest element. Around the base, tuck turmeric rhizomes where they get warmth and filtered light. Place a Camellia sinensis tea plant in its own pot in a shaded corner with acidic soil. Train Japanese knotweed up a small trellis in a fully sealed container isolated from any open ground. Give astragalus its own deep pot because the taproot grows straight down. Set a dwarf olive tree in a large container where it gets full sun. Hang a strawberry basket from the edge of your patio or balcony railing. On your kitchen counter, keep 3 mason jars in rotation, 1 sprouting broccoli seeds, 1 sprouting wheat berries, 1 resting and ready to restart. That indoor station requires no outdoor space at all. In your pantry, a bag of dried hibiscus calices sits ready for tea whenever you want it. 10 plants, 5 aging forces, every single one of them covered. Zombie cells are targeted by strawberry phycetine and turmeric curcumin. Shrinking telomeres are protected by green tea, EGCG, and rebuilt by astragalus cycloastragenol. The broken recycling system is reactivated by broccoli sprout sulforaphane and wheat germ spermidine through 2 independent pathways. Dying mitochondria are renewed by pomegranate urolithin A and shielded by olive leaf oleuropein. Sugar damage is intercepted by hibiscus anthocyanins, while Japanese knotweed resveratrol delivers systemic antioxidant defense across every front. Now picture what a monthly supply of those 10 compounds would cost in supplement form. Phycetin runs $40 to $60. TA 65 costs well over $100. Urolithin A adds another $50 to $70. Stack resveratrol, sulforaphane, curcumin, EGCG, oleuropein, spermidine, and hibiscus extract on top of all that. You are looking at $300 to $500 every single month for the rest of your life. Growing these whole foods at home is cost-effective compared to buying bottled extracts. Incorporating nutrient-rich plants like strawberries, broccoli sprouts, and wheat berries into your regular diet gives your body whole food compounds without the premium price tag of synthetic supplements. The reason you are hearing this from a video and not from the company selling you those compounds is that there is no profit margin in a plant that grows itself for free. 2 Nobel Prizes identified the mechanisms. Thousands of studies confirmed the compounds. 10 plants deliver them all. And 1 bucket holds everything. These plants do not care about your zip code or your income. They ask for soil, water, and sunlight. The answers were always right here, growing in plain sight, waiting for someone to finally look down. The scientist's name was George Wald. He worked at Harvard University for over 40 years. In the 1950s and 60s, he conducted a series of experiments that changed everything medicine understood about human eyesight. He proved that a molecule called retinol, which is derived from vitamin A and belongs to a family of pigments called carotenoids, is the molecule that allows photoreceptor cells in the retina to detect light. Without this pigment, you cannot see. The Nobel Committee in Stockholm awarded him the prize in physiology or medicine because his discovery was not theoretical. It was the physical mechanism of vision itself. But Wald's work opened a door that led somewhere the supplement industry would rather you did not follow. 2 specific carotenoids from that same pigment family called lutein and zeaxanthin concentrate in a tiny region of the retina called the macula. This region is only 2 millimeters wide. It sits at the exact center of your visual field. Every time you read a sentence, recognize a face, or thread a needle, you are using your macula. Lutein and zeaxanthin do 2 things there. They filter high-energy blue light before it damages the photoreceptors, and they neutralize free radicals that accumulate from a lifetime of light exposure. They are a filter and a shield built into the biology of your eye. Your body cannot manufacture lutein or zeaxanthin on its own. It must get them from food. And some of the richest food sources on Earth are plants you can grow in a bucket. When those pigments thin out, the macula breaks down. The clinical name for this is age-related macular degeneration. It is the leading cause of irreversible blindness in adults over 50 in the developed world. More than 11 million Americans have some form of it right now. That number is projected to reach 22 million by 2050. In 2006, the United States National Eye Institute launched the largest clinical trial ever conducted on carotenoids and eye health. They called it the Age-Related Eye Disease Study 2. Over 4,000 participants across 82 clinical sites in the United States enrolled. The trial ran for 6 years. The results were published in JAMA Ophthalmology in 2013. Daily supplementation with 10 milligrams of lutein and 2 milligrams of zeaxanthin reduced the risk of progression to advanced macular degeneration by 18 to 26% in participants who had low dietary intake of these pigments. This was not a fringe study in an obscure journal. This was the National Institutes of Health spending tens of millions of dollars to prove that these plant-derived pigments protect aging human eyes. So the science is settled. The Nobel Committee confirmed the mechanism. The National Eye Institute spent 6 years proving the application. But the plant that produces more of this compound than anything else on Earth is not in any pharmacy. It is not in any eye doctor's waiting room pamphlet. It is in the garden section of a hardware store, mislabeled as decoration. The plant is a marigold, Tagetes erecta, the tall, bright orange and yellow flower that gardeners buy in 6-packs every spring to border their tomato beds and keep aphids away. You have seen it 1,000 times. You may have planted it yourself last weekend. You spent $3 on a tray of them and pushed them into the soil next to your vegetables because someone told you they repel pests. What nobody told you is that this flower is the single largest commercial source of lutein and zeaxanthin supplements on Earth. Walk into any health food store in the United States tonight. Pick up a bottle of lutein supplements. Turn it over, read the label. If it says Flora GLO lutein, which is the most clinically studied lutein brand in the world, that lutein was extracted from marigold oleoresin. Cayman Industries, the company that manufactures Flora GLO, has built an entire global supply chain around Tagetes erecta flowers. They source marigold oleoresin primarily from contract farms across India in states like Andhra Pradesh, Karnataka, and Tamil Nadu. Hundreds of thousands of acres of marigold fields exist across southern India right now, not for weddings, not for decoration, but for the lutein extraction pipeline that feeds the Western supplement market. Here is how the chain works. Farmers in India grow the marigolds. They harvest the flower heads and dry them. Processing facilities extract the oleoresin from the dried petals using hexane solvent. The crude oleoresin is then shipped to purification plants where lutein is crystallized and standardized. The purified lutein is encapsulated in soft gel capsules and bottled under brand names. Those bottles are placed on shelves in pharmacies and health food stores across North America and Europe. The consumer pays $30 to $50 for a 30-day supply. Every step adds a company. Every company adds a margin. Extraction adds cost. Purification adds cost. Encapsulation adds cost. Branding adds cost. Shipping across oceans adds cost. Marketing the finished product adds cost. The raw material at the very beginning of this chain is a flower that grows in any warm climate from seed in 60 days and costs less than $3 for a packet of 50 seeds. The global lutein supplement market was valued at approximately $360 million in 2023. Industry analysts project it will exceed $500 million by 2030. Every dollar of that revenue depends on consumers not realizing that the active ingredient grows in a bucket on their porch. Now consider the science at the plant level. A single large Tagetes erecta flower head contains between 3 and 12 milligrams of lutein, depending on the cultivar and growing conditions. The AREDs2 study established the therapeutic dose at 10 milligrams per day. You spent $3 on a tray of them. But a massive warning here: never eat marigolds straight from a commercial nursery, as they are routinely drenched in toxic systemic pesticides meant for ornamental use. Instead, you must grow your own culinary-grade flowers from seed. Once grown cleanly, just a few dried or cooked petals can help match those clinical targets. A single plant produces dozens of flower heads over a growing season. A bucket with 3 marigold plants produces hundreds. The math is not complicated. The economics are devastating for anyone selling capsules. In Mexico, marigold petals have been used in cooking for centuries. They color rice. They flavor tamales. They appear in traditional Day of the Dead celebrations, not just as visual offerings, but as ingredients in food served to the living. Mexican grandmothers were putting lutein into their families' diets through marigold petals long before the supplement industry gave the molecule a brand name. In India, marigold garlands draped at temple entrances and around the necks of honored guests are not just spiritual. The flowers are also fed to poultry. Free-range chickens that eat marigold petals produce eggs with deep orange yolks. That orange color is lutein. Indian poultry farmers have known for generations that marigold in the feed makes the eggs richer. Western agriculture adopted this practice too, but replaced the whole flower with a synthetic additive and charged more for it. Growing marigolds in a container is one of the simplest tasks in all of gardening. Use a 5-gallon bucket with drainage holes drilled in the bottom. Fill it with standard potting mix. Plant 3 to 4 seeds per bucket, a 1/4 inch deep. Place the bucket in full sun at least 6 hours per day. Water when the top inch of soil is dry. Seeds germinate in 5 to 7 days at around 21 degrees Celsius. The plants flower within 60 to 90 days. Deadhead spent blooms by pinching them off and the plant produces more flowers. In warm climates, marigolds self-seed. Let a few flower heads dry on the plant at the end of the season. The seeds drop into the soil. Next spring, they return without replanting. For maximum lutein content, grow tall African marigold varieties. Crackerjack is widely available and produces large, dense flower heads. Antigua is another high-performing cultivar. These are not the small French marigolds sold as edging plants. These are the tall, robust varieties that reach 2 to 3 feet in height and produce flower heads the size of a tennis ball. Harvest the petals when the flowers are fully open. Spread them on a screen or paper towel in a warm, dry area out of direct sunlight. They dry in 2 to 3 days. Store dried petals in a sealed jar in a dark cupboard. Add them to soups, rice, scrambled eggs, smoothies, or tea. The flavor is mildly floral with a slight bitterness. The color is deep gold. The lutein is bioavailable when consumed with a small amount of dietary fat because lutein is fat soluble. One $3 seed packet, one $5 bucket, 60 days of patience. A supply of the exact same compound that the supplement industry extracts, purifies, encapsulates, ships across the planet, and sells for $40 a month. The plant was never hidden. The science was never secret. The marigold sat in every garden center in every town in every country, and nobody connected the flower in the pot to the capsule in the bottle. But marigold is the obvious answer once you see it. The next plant shocked even the researchers who study it. It costs more per gram than gold. It has been traded along the Silk Road for 3,000 years, and a clinical trial in Italy proved it can measurably improve macular function in patients who are already losing their vision. You can grow it in a wide pot on your patio for the cost of a single restaurant meal. The plant is saffron crocus, Crocus sativus. You know saffron as a spice. You associate it with paella in Spain, risotto in Italy, biryani in India. It is the most expensive spice on earth by weight. Retail prices range from $5 to $15 per gram, depending on grade and origin. In 2010, a researcher named Silvia Bisti and her colleagues at the University of L'Aquila in central Italy published a study that changed the conversation about botanical interventions for macular degeneration. They designed a randomized, double-blind, placebo-controlled clinical trial. This is the gold standard of medical evidence. They recruited patients diagnosed with early age-related macular degeneration. They gave the treatment group 20 milligrams of oral saffron supplementation daily for 90 days. They measured retinal flicker sensitivity, which is the eye's ability to detect rapid changes in light. This is one of the first measurable functions to degrade when the macula begins to fail. The results were statistically significant. Patients taking saffron showed meaningful improvement in retinal flicker sensitivity compared to the placebo group. The study was published in Investigative Ophthalmology and Visual Science, one of the most respected peer-reviewed journals in ophthalmology. This was not a blog post. This was not a wellness influencer's opinion. This was a controlled trial at a major European university published in a top-tier scientific journal. In 2012, Beastie and her team extended the study. They followed patients for 14 months on continuous saffron supplementation. The improvements were sustained. They were not a temporary spike that faded. The retinal function gains held steady across the entire extended observation period. Additional research groups took notice. Scientists at Tehran University of Medical Sciences conducted parallel investigations into saffron's active compounds. Australian researchers explored similar angles. The body of evidence grew quietly while the supplement industry continued marketing its existing product lines. 3 compounds in saffron are responsible. The first is crocin. Crocin is a carotenoid glycoside that gives saffron its deep red and gold color. It is a potent antioxidant. Saffron contains unique compounds that clinical trials show can support eye health. While crocin itself is actually broken down into crocin during digestion, this highly bioavailable metabolite easily crosses the blood-retinal barrier to protect your vision. Crocin gets through. It reaches the cells it needs to protect. The second compound is crocetine. Research published in Molecular Vision in 2015 demonstrated that crocetine has neuroprotective effects on retinal ganglion cells. These are the cells that transmit visual information from the retina to the brain through the optic nerve. Protecting them is protecting the wiring of your visual system. The third compound is safranol. This is the volatile molecule that gives saffron its distinctive aroma. Studies from Tehran University of Medical Sciences have documented anti-inflammatory properties of safranol in retinal tissue. Chronic low-grade inflammation in the retina is one of the drivers of macular degeneration progression. Safranol works against that driver. 3 compounds, 3 mechanisms, 1 spice that has been sitting in kitchen cabinets for 3,000 years. Now consider the economics. Saffron retails at $5 to $15 per gram. The therapeutic dose used in the Italian clinical trial was 20 milligrams per day. 20 milligrams is 2/100ths of a gram. Even at the highest retail price, 1 gram of saffron provides 50 daily doses. That is roughly 10 to 30 cents per day for a compound with published clinical trial evidence of macular function improvement. Walk into a supplement store. Find a bottle of saffron extract capsules. The price is $30 to $60 for a 30-day supply. The active ingredient inside those capsules is the same crocin and crocetine that exists in the raw spice. But the supplement company has extracted it, standardized it, encapsulated it, branded it, and marked it up by several 100%. The margin between the raw spice and the finished capsule is where the silence lives. But the real economic shift when you grow your own. 100 saffron crocus corms cost between $40 and $60 from a reputable nursery. Plant them in a wide, shallow container at least 15 centimeters deep. Use well-draining potting mix. Push each corm about 10 centimeters into the soil. Do this in late summer, August or September in the northern hemisphere. The corms flower in autumn, typically October or November. Each flower produces exactly 3 red stigmas. Those 3 stigmas are saffron. Harvest is simple. When the purple flowers open in the morning, pluck them. Pull out the 3 red thread-like stigmas with your fingers or tweezers. Set them on a warm, dry surface. A plate on a sunny windowsill works. They dry in 24 hours. Store dried stigmas in a small, airtight glass jar away from light and moisture. 100 corms produces approximately 1 gram of dried saffron in the first year. 1 gram provides 50 daily doses at the 20 milligram therapeutic level. That is nearly 2 months of supplementation from the first harvest alone. But here is where the compounding begins. Saffron corms multiply. Each corm produces 1 to 3 daughter corms annually. After 2 years, your 100 corms become 150 to 200. After 3 years, 200 to 300. After 5 years, you may have 500 or more corms in expanded containers. Your saffron supply compounds every single year, while the supplement subscription costs the same amount forever. The corms need one specific condition: a dry dormancy period in summer. Do not water the container from late May through late August. The corms rest underground. They need that dry, warm period to initiate flower bud formation. When you resume watering in September, the corms wake up and push their flowers through the soil surface within weeks. In USDA hardiness zones 6 through 9, saffron crocus grows reliably outdoors. In colder zones, bring the container into an unheated garage for winter and return it outside in spring. In Iran's Khorasan province, families have cultivated saffron for over 3,000 years. The harvest is a community event. Grandmothers, mothers, and children sit together separating stigmas from flowers. In Kashmir, saffron fields stretch across high-altitude meadows, and the harvest determines the economic survival of entire villages. In La Mancha, Spain, saffron carries a protected designation of origin, and farmers follow traditions that predate the Roman Empire. These cultures understood something that modern ophthalmology is only now confirming with controlled trials. The compounds in those red threads do something measurable inside the human eye. The clinical data exists. The mechanism is documented. The plant grows in a pot, 2 plants, one that produces the pigment your eyes need to filter blue light, one that clinical trials proved can measurably improve failing macular function. Both grow in containers on a balcony or porch. Both cost less in a single season than 1 month of the supplements designed to replace them. But there are 8 more that belong in this bucket garden. 5 of them are so common, you might already have them growing within walking distance of your kitchen. Goji berry, kale, spinach, Swiss chard, and carrots. 5 plants. Every one of them validated for the same carotenoid compounds that George Wald identified in 1967. Every one of them grows in a container. Every one of them you already know by name. And every one of them delivers lutein, zeaxanthin, or beta-carotene at concentrations that the supplement industry would prefer you never measured for yourself. But the last 3 plants in this bucket garden are the ones that carry the strangest stories. One grows in a bucket of water with no soil at all. One is an edible flower that most gardeners plant for beauty and never once think to eat. And one comes with a World War II legend that the British government kept classified for decades. Watercress is one of the oldest leafy greens consumed by humans. Roman soldiers carried it on long marches. Hippocrates built the world's first hospital on the Greek island of Kos, near a stream specifically so he could grow watercress for his patients. What makes watercress unusual for a container gardener interested in eye health is that it requires no soil. Fill a bucket with water, add a thin layer of gravel at the bottom. Plant watercress cuttings or seeds in a small net pot suspended at the waterline. Place the bucket in partial shade. Change the water weekly. Within 3 weeks, you are harvesting a green that contains meaningful concentrations of lutein, along with beta-carotene, vitamin C, and vitamin K. A bucket of water on a shaded porch producing eye health compounds with almost zero effort. The simplicity is the point. Nasturtium is the flower your grandmother planted along the fence because it looked cheerful and kept the beetles off the beans. The bright orange and red flowers cascade over pot edges, they trail down walls, they attract pollinators. What almost nobody mentions is that nasturtium flowers and leaves are edible and contain high concentrations of lutein. You planted them for decoration. You planted them for pest control. They were helping protect your eyes the entire time, and nobody told you. Grow trailing varieties in a hanging basket or a wide container. They need full sun and actually prefer poor soil. Overfeeding nasturtiums produces leaves at the expense of flowers. Keep them slightly hungry and they bloom relentlessly. Eat the flowers in salads. Add the peppery leaves to sandwiches. The lutein is right there in the petals you were already admiring. And then there is bilberry. This is where the story turns strange. During World War II, Royal Air Force pilots flew night bombing missions over occupied Europe. The missions required exceptional night vision. A story circulated that Royal Air Force pilots ate bilberry jam before flights to sharpen their vision in darkness. British intelligence promoted this narrative. They encouraged the idea that bilberries were the secret behind the pilots' remarkable accuracy during nighttime raids. Decades later, historians and researchers revisited the story. The real reason for the Royal Air Force's improved night accuracy was not bilberry jam. It was the development of airborne interception radar, a technology so secret that the British government needed a cover story to prevent the Germans from learning about it. Bilberry jam was the cover. The propaganda was deliberate, the legend stuck. But here is the twist that makes this story worth telling on a channel called Secrets Beneath Nature. The underlying science was not entirely wrong. Bilberries are among the richest known sources of anthocyanins, a class of flavonoid compounds that give the berries their deep blue-black color. Research published in the Journal of Agricultural and Food Chemistry has documented that anthocyanins accumulate in ocular tissues after oral consumption. Studies in molecular nutrition and food research have shown that anthocyanins support retinal blood flow and reduce oxidative stress in eye tissue. The British government chose bilberry as the cover story precisely because there was just enough plausible science to make it believable. Bilberry, Vaccinium myrtillus, is the wild European cousin of the North American blueberry. True bilberry can be difficult to source as nursery plants, but dwarf blueberry varieties are widely available and contain the same class of anthocyanin compounds. Varieties like Top Hat and Blueberry Burst grow well in containers filled with acidic potting mix. They need full sun and consistent moisture. A single dwarf blueberry bush in a large pot produces fruit for years. The anthocyanins in every handful of berries support the same retinal blood flow that made bilberry jam a plausible cover story for classified radar technology 80 years ago. Let that settle for a moment. A World War II intelligence operation chose this berry because the eye science was just credible enough to fool the enemy. The propaganda was fake. The biology was real. The science confirms what the Nobel Committee recognized in 1967. The pigments that help protect human vision can be found abundantly in plants. They have always been available in plants. The supplement industry built a $360 million market by extracting these pigments from flowers, concentrating them into capsules, and selling them back to people who could have grown the flowers themselves. 10 plants, 10 buckets on a porch, a lifetime supply of the exact compounds your eyes were designed to use. The industries profit from the distance between a garden and a pharmacy shelf. These plants were never hidden. The research was never classified. The Nobel Prize was announced on international television. We just stopped looking at what was growing at our feet. The buckets are waiting. Every cell in your body takes damage every single day. Oxygen does it, sunlight does it, stress does it, processed food does it. The molecules responsible are called free radicals, and they chip away at the membranes around your cells, the energy factories inside your cells, and the DNA that tells those cells how to repair themselves. Your body has defenses against this: enzymes, antioxidant compounds, cellular repair crews that work around the clock. But those defenses weaken with age. As we age, our bodies experience a steady decline in endogenous antioxidant enzymes like superoxide, dismutase, and catalase, leaving our cells progressively more vulnerable to oxidative stress. This is not disease. This is the baseline cost of getting older. The supplement industry understood this decades ago. They built a global market around it worth over $400 billion annually. Vitamin C in bottles, vitamin E in soft gels, glutathione in IV bags at wellness clinics charging $200 per session, adaptogen powders sealed in foil pouches at $40 per bag. Every one of those products requires extraction, processing, encapsulation, branding, cold chain shipping, retail markup. Every one of those products generates repeat purchases month after month after month. But 5 plants exist that provide the same compounds those bottles try to replicate. All 5 are container-friendly. All 5 have been grown within arm's reach of the kitchen door by traditional cultures for centuries. Combined, They cover polyphenols and omega-3 fatty acids and adaptogens and glutathione and the vitamins C and E that drive your cellular repair systems. No extraction facility, no capsule machine, no supply chain. You grow them, you harvest them, you eat them. The first one on this list is something you have almost certainly pulled out of your own garden. You may have sprayed it, you may have cursed it, you may have thrown it into the compost bin without thinking twice. And it contains more cellular defense compounds per leaf than almost anything you have ever purchased in a bottle. It grows in sidewalk cracks. It grows between the stones on your patio. It pushes through gravel at the edge of your driveway, and it spreads across bare soil faster than you can pull it. You know it when you see it. Fat, reddish stems close to the ground. Small, thick leaves that look almost succulent. It appears every summer uninvited, and most gardeners in the Western world treat it as the enemy. This is purslane, Portulaca oleracea, and it has been quietly producing some of the most powerful cellular defense compounds on Earth while you are pulling it out and throwing it away. Start with the omega-3 content. Purslane contains the highest concentration of alpha-linolenic acid ever measured in any leafy green vegetable. Alpha-linolenic acid is the plant form of omega-3 fatty acids, the same category of compound that the supplement industry sells as fish oil capsules and krill oil and algae soft gels. Purslane produces it for free in the crack of your driveway. No fishing vessel required, no processing plant, no capsule, no bottle, no shelf, just a leaf. But the omega-3 is not the reason purslane belongs in your cellular defense bucket. Purslane contains glutathione. This is the compound your cells use as their primary internal antioxidant. Your liver manufactures glutathione constantly. It is the frontline defender inside every cell, neutralizing the free radicals that damage membranes and mitochondria. By age 60, your body's natural glutathione production has dropped significantly. The supplement industry noticed. Glutathione capsules now sell for $30 to $60 per bottle. Liposomal glutathione formulations cost more. IV glutathione drips at aging clinics cost $200 per session. Purslane contains measurable levels of this same compound, and it grows in gravel. There is another layer. Purslane also contains melatonin at concentrations 10 to 20 times higher than most fruits and vegetables. Most people know melatonin as the sleep supplement sold at every pharmacy checkout counter. But melatonin is not just a sleep molecule. Inside the cell, it functions as a direct free radical scavenger. It crosses barriers that other antioxidant compounds cannot cross. It reaches the mitochondria, where the heaviest oxidative damage occurs during aging. The same compound people buy in 3 milligram tablets at the drugstore grows in the cracks of the parking lot outside that drugstore. Now consider who already knew this. In Greece, families have eaten purslane for centuries. They call it androcla or glystrida and serve it raw in salads with olive oil and feta cheese and fresh tomatoes. Walk through a village market on any Greek island in summer and you will see bundles of it stacked next to the lettuce. In Mexico, they call it verdolaga and stew it with pork and tomatillos and fresh chilies. Street vendors in Mexico City sell verdolaga tacos from metal carts on the sidewalk. In Turkey, purslane has appeared in meze spreads for generations. In India, village markets sell fresh bundles of it every morning before the heat arrives. Across every continent except Antarctica, someone's grandmother knew exactly what this plant was worth. No seed company promotes purslane. It self-seeds so aggressively that one planting creates a permanent supply. No supplement company can patent its glutathione because it is not engineered. It is not branded, it is not formulated, it simply grows wherever it is not killed. And for most of the Western world, it is killed on site. Growing purslane requires almost nothing. Full sun, minimal water, warm soil. It thrives in the shallow edges of a container planter where other plants would struggle. Let one plant flower and set seed, and you will never need to purchase purslane seed again. It returns every season without permission, like a guest you stopped inviting who keeps showing up with exactly what you needed. Purslane handles the omega-3 and the glutathione and the melatonin. But there is another compound your aging cells need even more urgently: the polyphenols and the vitamin concentrations that directly protect your cell membranes from oxidative breakdown. And the plant that delivers them grows on the other side of the world in quantities that make every supplement bottle on the shelf look like a rounding error. In the dry zones of northern India, families have grown a particular tree outside the kitchen door for as long as anyone in the village can remember. They call it sajan in Hindi. In the southern states of Tamil Nadu and Kerala, they call it Murangai. Travel west to the Sahel region of sub-Saharan Africa, and you will find the same tree growing in village compounds from Senegal to Niger to Chad. In parts of West Africa, they call it nebedaye. Some local translations render this loosely as never die. The English name is moringa. The botanical name is Moringa oleifera. And gram for gram, its leaves contain more protective compounds than almost any single food source ever analyzed. Start with the polyphenols: quercetin, chlorogenic acid, beta-carotene, camphorol. These are the specific compounds that neutralize the free radicals attacking your cell membranes, your mitochondrial walls, and your DNA repair enzymes. Moringa leaves contain all of them. Not in trace amounts, in concentrations that rival or exceed the most celebrated antioxidant foods on Earth. Blueberries get the marketing. Moringa gets the chemistry. The vitamin profile is staggering. Dried moringa leaf powder contains vitamin C at roughly 7 times the concentration of oranges by weight. Vitamin E levels approach those found in sunflower seeds. These are the 2 vitamins most directly involved in protecting the lipid membranes that surround every cell in your body. Vitamin C works on the water-soluble side. Vitamin E works on the fat-soluble side. Together, they form a 2-layer shield. The supplement industry sells them in separate bottles at separate prices on separate shelves. Moringa delivers both from the same leaf. The leaves also contain compounds called isothiocyanate. These suppress the inflammatory pathways that your cells activate under chronic stress, the same pathways that over decades contribute to the slow deterioration of joints and blood vessels and organ tissue. Traditional Ayurvedic practitioners in India prescribe moringa leaf preparations for exactly these conditions. They called the tree shigru. They documented its properties in texts that predate modern chemistry by more than 1,000 years. They did not know what an isothiocyanate was. They observed the outcome across generations of patients and recorded what worked. Here is where the economics become impossible to ignore. In the 1990s and 2000s, international aid organizations began distributing moringa seeds and seedlings across sub-Saharan Africa and South Asia. The World Health Organization supported it. UNICEF promoted it. Dozens of national governments funded planting programs. They planted moringa in schoolyards in Senegal. They grew it in refugee camps in Bangladesh. They established moringa nurseries in rural Niger. The explicit purpose was to combat malnutrition because a single moringa tree could provide enough leaf nutrition to supplement an entire family's diet for years. This was not alternative medicine. This was not a wellness trend. This was the global public health establishment acknowledging that one tree could replace a cabinet full of supplements for populations that could not afford bottles, while aid workers were planting moringa trees in village commons across 3 continents. Western consumers were buying individual vitamin C tablets in plastic containers at the pharmacy checkout. No major supplement company funded moringa research in the West during those decades. No pharmaceutical company developed a moringa-based product line. No grocery chain in North America stocked fresh moringa leaves, a tree that produces harvestable leaves within 60 days of planting, a tree that tolerates drought and poor soil, a tree that non-governmental organizations considered important enough to distribute by the millions. And it remained invisible in the country that spends more on dietary supplements than any other nation on Earth. The pattern is not conspiracy, it is structure. A tree you plant once does not generate repeat seed sales. A leaf you harvest from your porch does not generate retail revenue. A food that replaces 3 supplement bottles simultaneously does not generate 3 separate revenue streams. Moringa fell into the same structural gap that every self-sustaining crop falls into. The systems that promote profitable products simply had no mechanism to promote a product that eliminates the need for products. Growing moringa in a container requires a deep pot, at least 18 inches of soil depth because the taproot drives downward fast. Standard potting mix with added perlite for drainage. Full sun, moderate water during establishment, then less as the root system matures. In warm climates, it becomes a perennial that produces year after year. In cooler climates, treat it as a vigorous annual or bring it indoors before frost. Either way, you will have harvestable leaves within 2 months of planting. Harvest often. The more you cut, the more it branches. The more it branches, the more leaves it produces. Moringa floods your cells with the polyphenols and the vitamins. Purslane covers the omega-3 and the glutathione and the melatonin, but there is a third layer of cellular defense that neither of those plants provides, the structural repair layer, the collagen and the connective tissue and the neural protection that hold your body's architecture together as the decades pass. And the next plant on this list has been called the herb of longevity for over 2,000 years for exactly that reason. Across South and Southeast Asia, one small creeping plant has earned a name that no other herb carries. In Sri Lanka, they call it Gotu kola. In the Ayurvedic medical tradition of India, it is classified as a Medhaya Rasayana. This translates roughly as a rejuvenator of the mind. In Chinese traditional medicine, it appears under the name Ji Shui Kao. In Malaysia and Indonesia, they call it Pegaga. But the name that matters most is the one ordinary people gave it across a dozen countries and more than 100 generations, the longevity herb. In Sri Lanka, there is a folk observation that has been passed between families for centuries. Elephants eat Gotu kola. Elephants live long. Elephants remember. Sri Lankan herbalists watched the animals graze on this low-creeping plant in forest clearings and along riverbanks, and they drew a simple connection. The same plant that the longest-lived land animal seeks out might be worth eating yourself. No controlled trial produced this insight. No laboratory confirmed it. Generations of watching and correlating produced it. And then modern research arrived and found something remarkable. The traditional observers were not wrong. Gotu kola contains compounds called triterpenoid saponins. The 2 most studied are asiaticoside and madecassoside. These compounds stimulate the production of type 1 collagen in human tissue. Collagen is not a cosmetic molecule despite what the beauty industry has done with the word. It is the structural protein that holds together your skin, your blood vessel walls, your tendons, the lining of your digestive tract, and the connective tissue between your organs. After 50, your body produces less of it every year. The decline is measurable. The consequences accumulate. Joints stiffen, skin thins, blood vessels lose elasticity, wound healing slows. The supplement industry responded with collagen peptide powders sold in tubs for $35 to $50 each. These products are derived from bovine hide or fish scales. They are processed, hydrolyzed, flavored, and packaged. Gotu kola does not deliver collagen from outside. It encourages your own cells to produce more of it from inside. The mechanism is fundamentally different. One approach replaces from the outside, the other restores the factory on the inside. Research on healthy elderly volunteers found that daily consumption of Gotu kola over a period of 2 months improved working memory and cognitive alertness compared to baseline measurements. The traditional classification as a brain herb aligns with what the clinical observations now support. The compounds appear to improve microcirculation to the small blood vessels that feed neural tissue and reduce oxidative damage in the brain. Sri Lankan families who fed Gotu kola to their elders were not guessing. They were acting on accumulated observation that modern instruments eventually validated. Walk through a market in Colombo or Kandy in Sri Lanka, and you will see fresh Gotu kola leaves bundled and stacked in piles. In Indonesia, the leaves appear in fresh salads called pasel and gado-gado, alongside peanut sauce and steamed vegetables. In Vietnam, street vendors press the leaves into fresh juice with green apple and honey and sell it cold from metal carts. In southern India, chutneys made from fresh Gotu kola leaves are served alongside rice and dal at family meals. Hundreds of millions of people consume this plant weekly. In the average American grocery store, it does not exist. Growing Gotu kola requires moisture and patience. It is a creeping ground cover plant that spreads along the soil surface by runners. In a container planter, it fills the top layer beautifully. It prefers partial shade and consistently damp soil. Keep it moist and it spreads. Let it dry out and it sulks. In a mixed bucket with moringa and the other plants, Gotu kola occupies the surface layer around the base of the taller plants. It acts as a living mulch that holds moisture in the soil while simultaneously producing leaves that help protect your connective tissue and your neurons. Every square inch of soil surface it covers is doing double duty. Gotu kola handles the structural layer, the collagen, the connective tissue, the brain. But there is one more dimension of cellular defense that none of these three plants address: the hormonal stress response. The cortisol that floods your system under chronic pressure and accelerates cellular aging from the inside out. The next plant on this list was considered so sacred in one of the oldest medical traditions on Earth that Indian families planted it at their front door and blessed it every morning. In India, more than 100 million households keep a particular plant at the entrance to their home. Not in the garden behind the house, not on the kitchen windowsill, at the front door, in a clay pot or a stone planter or a raised bed built into the threshold itself. The plant is Tulasi, Ocimum tenuiflorum, holy basil. The tradition stretches back thousands of years and it persists in modern India today. Drive through any residential neighborhood in Chennai or Varanasi or Jaipur and you will see them. Tulsi plants standing guard at every other doorway. In Ayurveda, Tulsi carries the title Queen of Herbs. Families do not keep it at the entrance for decoration. They keep it there because it is the first medicine you pass on your way inside and the last one you pass on your way out. Morning prayers often include the Tulsi plant. Water poured over its leaves is considered purified. This is not superstition dressed as science. This is a culture that spent 3,000 years observing which plants kept people healthier and decided this one deserved the front door. Here is what modern analysis found when it finally looked closely. When your body produces cortisol under chronic stress, that hormone does not just make you feel anxious or disrupt your sleep. Cortisol accelerates wear at the cellular level. It increases oxidative damage inside the mitochondria. It impairs the immune system's ability to clear damaged cells. It disrupts the chemical signals that coordinate cellular repair. Sustained over years and decades, chronic cortisol elevation creates a biochemical environment inside your cells that mimics and magnifies the effects of aging itself. Stress and aging use the same damage pathway. They are not separate problems. They are the same problem arriving from different directions. Tulsi is classified as an adaptogen. This means it does not push the body in one fixed direction the way a pharmaceutical drug does. It modulates. If cortisol runs too high, Tulsi helps bring it down. If the stress response becomes erratic, Tulsi helps stabilize it. The active compounds include eugenol, the same molecule found in clove oil that functions as an anti-inflammatory agent; rosmarinic acid, the same compound found in rosemary that reduces allergic and inflammatory responses; ursolic acid, a compound that supports cellular integrity under stress; apigenin, a flavonoid that calms without sedating. These compounds work across multiple pathways simultaneously. This is not a single target intervention. This is a plant that addresses the system. In Thailand, holy basil appears in the single most popular street food dish in the country, Pad Kra Pao. Stir-fried minced meat with holy basil leaves, garlic, chilies, and fish sauce served over rice with a fried egg on top. Walk down any street in Bangkok, and the smell of holy basil hitting a hot wok will find you before you find the vendor. In traditional medicine practices across South and Southeast Asia, tulsi preparations have treated coughs and digestive complaints and skin conditions and fevers for longer than written records can confirm. The knowledge base spans continents and centuries. Growing tulsi is almost effortless. It is one of the most enthusiastic container plants in the herb world. Full sun, warm conditions, regular harvesting. The more you pinch the growing tips, the bushier it becomes. It self-seeds generously in warm climates. You can harvest tulsi leaves multiple times per season from the same plant. In a mixed container planter, tulsi occupies the mid-height zone, taller than the creeping go-to cola at the soil surface, shorter than the moringa reaching upward from the center. It fills the middle layer of your cellular defense garden, and it does so happily for the entire growing season. Purslane, moringa, go-to cola, tulsi, 4 plants covering 4 layers of cellular defense, omega-3 and glutathione from the ground level, polyphenols and vitamins from the canopy, structural collagen support from the creeping surface, hormonal stress modulation from the middle tier. But there is a 5th plant that works slower and works deeper and addresses the one thing none of the other 4 can touch. It is not a leaf, it is a root. And it has been part of the oldest rejuvenation tradition in recorded medicine for over 3,000 years. The name ashwagandha comes from Sanskrit. It translates roughly as smell of the horse. Not because the plant smells like a stable, although the root does carry a strong, earthy odor that surprises people the first time. The name implies that consuming the root gives you the vitality and endurance of a horse. In the Ayurvedic tradition, ashwagandha belongs to the Rasayana class of herbs. Rasayana does not translate to anti-aging in the way Western supplement marketing uses that phrase. It means restoring the body's own capacity to repair and defend itself, rebuilding the foundation rather than repainting the walls. The active compounds in the root are called withanolides. These are steroidal lactones that modulate the HPA axis, the communication loop between your brain and your adrenal glands that controls cortisol output. Research on adults taking ashwagandha root extract daily for 60 days measured cortisol levels reduced by approximately 28% compared to placebo. 28%. Consider what that means over years and decades. If chronic cortisol elevation is one of the primary biochemical accelerators of cellular aging, then reducing it by nearly a third does not reverse time, but it removes one of the heaviest weights pressing down on your cellular repair systems. There is a deeper layer to the ashwagandha story. Pilot research has measured the activity of an enzyme called telomerase in healthy volunteers taking ashwagandha. Telomerase maintains the protective caps on the ends of your chromosomes. These caps are called telomeres, and they shorten each time a cell divides. When they get too short, the cell can no longer divide properly. This process is one of the most directly measurable markers of cellular aging. Over the last several years, rigorous peer-reviewed clinical trials have directly evaluated ashwagandha's impact on aging markers, showing measurable improvements in telomerase activity and cellular stress biomarkers. But the direction is consistent with what Ayurvedic Rasayana practitioners documented through 3,000 years of clinical observation without ever knowing what a telomere was. They saw the outcomes in their patients. Modern instruments now measure one possible mechanism behind those outcomes. Every other plant in this bucket is harvested from the leaf. Ashwagandha is harvested from the root. This changes the timeline. You do not harvest ashwagandha in 60 days, you wait months. You let the root develop density and concentration underground where you cannot see it working. The withanolides accumulate in the root tissue slowly, building potency over time. This is the patient plant in the lineup, the one that rewards waiting, the one that teaches you that some forms of cellular defense cannot be rushed. Ashwagandha needs depth and drainage. sandy, well-draining soil in a deep pot. At minimum 12 inches, ideally 18 inches, full sun and infrequent watering once established. It does not like wet feet. Overwatering kills ashwagandha faster than neglect does. In a mixed container arrangement, ashwagandha occupies its own deep companion pot beside the main planter. Separate root space, shared growing station. It stands next to the main bucket, like a quiet partner doing its work underground while the leafy plants above do theirs in the sun. Now picture the arrangement. One wide, deep planter, at least 20 inches across and 18 inches deep, sitting on your porch or your patio or a sunny patch of balcony. While you can arrange these 5 plants together on a sunny patio or balcony to create a unified cellular defense station, you'll want to give them separate pots tailored to their wildly different personalities. And beside the main planter sits the deep companion pot where ashwagandha develops its root in silence, building the withanolide concentration that supports your cells at the chromosomal level. 5 plants, 1 growing station, polyphenols and vitamins C and E from the moringa canopy, omega-3 fatty acids and glutathione and melatonin from the purslane at the edges, collagen stimulation and neural protection from the go-to cola at the surface, adaptogenic stress modulation from the tulsi in the middle, deep cellular repair support from the ashwagandha root below ground, 5 layers of defense that the supplement industry sells in 5 separate bottles from 5 separate companies at 5 separate price points. Standard potting mix with added perlite for the moringa zone, a slightly richer moisture-retentive blend around the go-to cola, drainage holes in every container, water when the top inch of soil dries out for the main planter, water less for the ashwagandha pot. Harvest the moringa and tulsi and purslane leaves regularly throughout the season. Let the go-to cola spread. Let the ashwagandha root develop on its own schedule. The system requires less maintenance than a window box of petunias and produces more cellular defense compounds than a shelf full of bottles. The supplement industry built a $400 billion global market selling isolated compounds in capsules extracted from plants that anyone with a bucket and some soil could grow at home. The vitamin C came from plants. The vitamin E came from plants. The omega-3 came from organisms that ate plants. The glutathione precursors came from plants. The adaptogenic compounds came from roots and leaves that traditional families grew outside the kitchen door for centuries before anyone invented a capsule. Nobody suppressed these 5 plants. That is not what happened. What happened is simpler and in some ways harder to accept. No corporation profits when you grow your own cellular defense system. No quarterly earnings improve when a weed replaces a supplement. No marketing budget exists for a plant that self-seeds in sidewalk cracks and asks nothing from you in return. The system that promotes profitable products simply has no mechanism to promote products that eliminate the need for products. But the plants do not care about profit margins. Moringa has grown in Indian dooryard gardens since before the pharmaceutical industry existed. Tulsi has stood at the front entrance of Indian homes since before the supplement aisle was invented. Purslane has pushed through sidewalk cracks in every warm climate on Earth since before anyone trademarked the word antioxidant. Go-to cola has crept across forest floors in Sri Lanka since before the collagen peptide industry was born. Ashwagandha has developed its root in dry Indian soil since before anyone measured a telomere. 5 plants, 1 bucket, they were always there. Nobody had a reason to tell you. 2,000 years ago in central China, physicians began writing down everything they knew about plants. They tested roots, leaves, bark, and seeds on themselves and on patients across generations. They classified hundreds of plants into 3 tiers. The lowest tier treated symptoms, the middle tier treated disease, and the highest tier, a category reserved for fewer than a dozen plants in the entire system, was called superior. A superior herb could be taken every single day for an entire human lifetime without causing harm. It did not treat illness. It prevented the body from breaking down in the first place. 1 root earned that rank above almost all others. The oldest surviving herbal text in human history is called the Shenong Ben Cao Jing. It was compiled around 200 AD in China. Inside its pages, this root appears with a specific description. It strengthens what Chinese physicians called Wei Qi, the body's protective energy. Not a metaphor, not a spiritual concept. A functional description of what modern science would eventually call immune defense. 2,000 years before anyone understood white blood cells or T cells or natural killer cells, Chinese doctors had identified a root that reinforced the body's ability to fight off disease, and they wrote it down. During the Tang Dynasty, which ruled China from 618 AD to 907 AD, imperial physicians traveled with military campaigns. Records from that period describe this root being given to soldiers before long marches, not to treat wounds, not to numb pain, for endurance, for the ability to keep the body functioning under extreme stress when food was scarce and rest was impossible. The military archives survive. The root's reputation was not folklore, it was state policy. Across the Mongolian steppe, herders carried dried slices of this root in leather pouches during winter migrations. Temperatures dropped below freezing for months, animals died, supplies ran out. The herders chewed thin slices of the root as they walked. It did not feed them, it kept their bodies from collapsing under conditions that should have broken them. The tradition continued for centuries. Families passed the knowledge from one generation to the next without ever needing to explain the chemistry behind it. The root worked, that was enough. The root also traveled along the ancient Silk Road. Traders moving between Xi'an and Samarkand packed dried bundles of it alongside silk, tea, and spices. Merchants did not carry dead weight across deserts and mountain passes. Everything on a camel's back had to earn its place. This root earned it. Traders chewed it to maintain stamina during weeks of walking through terrain that broke men and animals alike. At trading posts along the route, it changed hands alongside goods worth fortunes. But unlike silk, the root could be replanted at the destination. Unlike gold, it multiplied in the ground. Wherever the Silk Road touched, the root followed. And wherever it was planted, it stayed. Now here is where the Western world lost the thread. In Mandarin, this root is called Huang Qi. It means yellow leader. Yellow for the bright golden color of the inner root when sliced open. Leader because it commands and strengthens the body's energy systems. 500 million people across China know this name the way Americans know the word aspirin. It sits in glass jars in every traditional medicine shop from Beijing to Guangzhou. You can buy a bag of it for $2. In Japan, traditional medicine is called Kampo. It has been practiced for over 1,000 years, and it is still integrated into the modern Japanese healthcare system. Inside Kampo formulas, this root appears under the name Oji. Japanese physicians prescribe it for fatigue, for immune support, for recovery after illness, and here is what matters. Japan's national health insurance system covers Kampo treatments. This root is one of the herbs included in those covered formulas. A government looked at the evidence and decided it was real enough to pay for. In Korea, the root is called hwangji. Korean grandmothers brew it into a soup called hwanggi samgyeetang. It is chicken soup with the root simmered inside for hours. Families serve it during winter to protect children from getting sick. They serve it to elderly parents to keep them strong. Walk into any traditional market in Seoul and you will see hwangi bundled in plastic bags stacked next to ginseng. It is not exotic, it is not hidden, it is groceries. The tradition runs deeper than soup. In rural Korean villages, families keep bundles of dried hwangi root hanging in the kitchen the way Western households keep a first aid kit in the bathroom. When a child comes home from school with a cough, the grandmother reaches for the root before she reaches for anything else. When a new mother is recovering after childbirth, the family prepares hwangi broth and brings it to her bedside every morning for weeks. The root is not a supplement in Korea. It is not a trend. It is infrastructure. It is woven into the rhythm of daily life so deeply that asking a Korean family why they use it is like asking an American family why they keep salt in the kitchen. The answer is too obvious to require an explanation. 4 languages, 4 names, and 1 root. And the exact same problem that buried other Asian plants in the West buried this one too. When Western databases could not unify the name, the research fractured across languages that nobody in American medicine was reading. A study published in Mandarin did not appear in English search results. A clinical observation documented in Japanese reached no American physician. The root had thousands of years of evidence behind it, but the evidence existed on the other side of a wall built from language and indifference. In the 1970s, Soviet scientists were studying a category of plants they called adaptogens. These were plants that helped the human body resist physical and psychological stress without side effects. The Soviet military funded this research because they wanted soldiers and cosmonauts who could endure extreme conditions longer. During that research, Soviet teams looked at populations in northern China and Mongolia who consumed this root as part of their daily diet. They noticed something unexpected. These populations showed cellular markers that did not match their chronological age. Their cells looked younger than they should have. The Soviet findings were published in Russian-language journals. They were not classified, but they were not translated either. Western researchers did not read Russian pharmacology journals in the 1970s. The Cold War did not just divide politics, it divided medicine. And a discovery about cellular aging sat in a language that American scientists never opened. The plant is Astragalus membranaceus, a legume, a member of the same family as beans and clover. It fixes nitrogen in the soil, meaning it feeds the ground it grows in rather than depleting it. In the wild, it looks like nothing special. Thin stems, small compound leaves, clusters of pale yellow flowers that most people would walk past without a second glance. But the root is where everything hides. Pull one from the ground after 4 years of growth and slice it open. The interior is bright yellow and fibrous. It smells faintly sweet. This unassuming root is what Chinese emperors, Mongolian herders, Japanese physicians, and Korean grandmothers have relied on for 2 millennia. The plant grows wild across a vast stretch of the northern hemisphere. From the grasslands of Inner Mongolia to the volcanic hillsides of Hokkaido, from the dry mountain slopes of Shanxi province to the rocky meadows of the Korean peninsula, it thrives in places most crops refuse to grow. Roadsides, gravel banks, thin soil over limestone, abandoned fields where nothing else takes hold. Farmers across Asia have always known it as the plant that shows up uninvited and never leaves. They did not need to cultivate it. They needed only to recognize it and harvest what the land offered freely. Modern in vitro and animal studies suggest that polysaccharides in Astragalus can interact with immune cells, showing potential to modulate immune responses. While preliminary clinical trials have explored these effects, high-quality, large-scale human data confirming systemic immune reinforcement remains limited. The root does what the ancient texts said it did. The only difference is that now there are lab measurements to prove it. The root also contains over 40 compounds called saponins. These are molecules that reduce inflammation through pathways that Western pharmaceutical companies have spent billions trying to replicate with synthetic drugs. Astragalus does it with chemistry that evolved over millions of years. The root has been doing this work quietly since before humans had a word for inflammation. But the immune support and the anti-inflammatory effects were not what caught the attention of Western biotech companies. Something else did. Something that connected this ancient root to the single most important discovery in the science of aging in the last 50 years. In 2009, 3 scientists named Elizabeth Blackburn, Carol Greider, and Jack Szostak won the Nobel Prize in Physiology or Medicine. Their discovery changed everything we understand about why the human body ages. They identified structures called telomeres. Telomeres are protective caps that sit on the ends of your chromosomes. Think of the plastic tips on the ends of shoelaces. They prevent the lace from fraying. Telomeres do the same thing for your DNA. Every time a cell divides, the telomeres shorten slightly. Over years and decades, they get shorter and shorter. When they become too short, the cell can no longer divide. It becomes what scientists call senescent. It stops functioning. It ages. It dies. This is cellular aging at its most basic level. The clock inside your cells is your telomere length, and it has been counting down since the day you were born. The Nobel team also identified an enzyme called telomerase. This enzyme can rebuild telomeres. It can add length back to the caps. It can, in a very real sense, reset the clock. But in most adult human cells, telomerase is nearly silent. The gene that codes for it exists in every cell, but the enzyme barely activates. Cells divide, telomeres shorten, aging continues. That is the default, unless something turns the enzyme back on. In the years following the Nobel Prize, research teams began screening natural compounds to find anything that could activate telomerase in human cells. They tested hundreds of plant extracts from dozens of countries. One compound activated telomerase more consistently and more potently than nearly anything else in the screening. That compound is called cycloastragenol, and it is found almost exclusively in one place on Earth, the root of Astragalus membranaceus. A closely related compound called astragaloside 4 showed similar telomerase activating properties. Both compounds concentrate in the root. Both are extracted through a process that traditional Asian medicine has been performing for 2,000 years, boiling the root in water, simmering it into tea, cooking it into soup. The grandmother standing in a kitchen in Seoul making hwangi samgyeetang for her grandchildren was extracting telomerase-activating compounds without ever knowing the word telomerase. The science caught up to the tradition. It took 2 millennia and a Nobel Prize to get there. Think about what that means in practical terms. Every time you see a wrinkle forming in the mirror, that is telomere shortening made visible. Every time a wound heals slower than it did 10 years ago, that is your cells dividing less efficiently because the protective caps are wearing thin. Every time your energy drops in the afternoon when it never used to, that is cellular aging expressing itself through fatigue. The clock does not stop, but the enzyme that rebuilds the clock exists inside you right now. It is just waiting for activation. And for thousands of years, people across Asia have been consuming the one root that provides that activation with every bowl of soup and every cup of tea. They did not call it anti-aging, they called it staying strong. The result was the same. What the research also revealed is that telomere length does not decline at a fixed rate for everyone. It varies, and the variance is significant. 2 people born the same year can have measurably different telomere lengths by middle age. Scientists tracking these differences found consistent patterns. Chronic stress accelerates shortening, poor sleep accelerates it, processed diets accelerate it, sedentary living accelerates it. These are not surprises, but the other side of that equation matters just as much. Certain inputs slow the shortening. In specific cell culture models, cycloastragenol has been shown to upregulate telomerase activity, which can help slow telomere loss or temporarily maintain length in vitro. However, clinical studies demonstrating that oral consumption of the raw root systematically rebuilds human telomeres or reverses systemic aging are not robustly established. The research on cycloastragenol specifically looked at what happens inside human immune cells after consistent exposure to the compound. T cells, the white blood cells responsible for coordinating the body's immune response, were the primary focus. T cells divide rapidly when the body faces infection. Rapid division means rapid telomere shortening. Old T cells with very short telomeres cannot respond to new threats the way young T cells can. This is one of the core reasons the immune system weakens with age. The soldiers become too old to fight effectively. What the studies found is that cycloastragenol appeared to maintain telomere length in these cells, preserving their ability to divide and respond. The immune system did not just function better in the short term, its capacity to function was being protected at the chromosomal level. This is not a metaphor. This is not a theory borrowed from ancient texts and dressed in modern language. This is what peer-reviewed cellular biology found when it finally looked at the root that Asian physicians had classified as superior 2,000 years earlier. The mechanism those physicians could not name, they had already identified through generations of careful observation. They saw the outcome and recorded it faithfully. The outcome was a body that resisted breakdown. The mechanism, it turned out, was running all the way down to the tips of the chromosomes. And there is one more layer that rarely gets discussed. Telomere shortening does not happen in isolation. When cells become senescent, they do not simply go quiet. They release a cascade of inflammatory signals into the surrounding tissue. Scientists call this the senescence-associated secretory phenotype. It is a kind of biological alarm that damaged cells broadcast to their neighbors. Over time, as more and more cells reach this state, the alarm becomes chronic. The inflammation becomes systemic. Chronic systemic inflammation is not just a symptom of aging. It is one of the primary drivers of every major age-related disease on the list. Heart disease, neurodegeneration, cancer, metabolic disorders. The connections run through inflammation, and inflammation runs in part through telomere biology. Researchers are investigating whether compounds that influence telomere biology might also interact with the cellular inflammatory cascade associated with aging. The 2,000-year-old classification of this plant as superior was not poetic, it was precise. Now here is where the story turns. Within 2 years of that Nobel Prize, a company in New York began aggressively marketing a supplement called TA65. The active ingredient was cycloastrogenol, extracted from Astragalus membranaceus root. The initial price was $600 for a 90-day supply. The marketing materials referenced telomere science, the packaging looked pharmaceutical, the branding suggested cutting-edge biotechnology. The source material was a root that Asian herb shops had been selling in open bins for $2 to $5 a bag for as long as anyone could remember. By 2023, the global anti-aging supplement market reached $62 billion. Products targeting telomere health occupy the premium tier of that market. Branded astragalus extracts from various companies sell for $100 to $300 per bottle. Some boutique formulations cost even more. The raw root, the exact same root containing the exact same compounds, sells in bulk on Chinese and Korean wholesale markets for pennies per gram. The price difference is not explained by the cost of extraction. You do not need a pharmaceutical lab to get cycloastrogenol out of astragalus root. While traditional water extraction pulls water-soluble polysaccharides into liquid form, free cycloastrogenol is present in the raw root in only trace amounts. Isolating or manufacturing bioavailable telomerase activating compounds typically requires specialized chemical extraction or hydrolysis techniques rather than standard kitchen brewing. The only difference is that one version built a corporation and the other built a tradition. If you search the major medical research databases for astragalus and telomerase, you will find studies. But compare the volume of that research to the volume of studies on patented anti-aging compounds, and the gap is staggering. The funding disparity follows the same pattern that appears with every plant that cannot be owned. Astragalus research comes overwhelmingly from institutions in China, Korea, and Japan, funded by governments, and published in languages that Western databases historically under-indexed. Western pharmaceutical money flows toward molecules that can be patented. A root that grows wild across half of Asia cannot be patented. A tea that a grandmother brews cannot be licensed. So the funding goes elsewhere. The research follows the funding, and the knowledge gap persists. Consider what happened with collagen supplements. For decades, East Asian cultures consumed slow-simmered bone broths and pigskin soups because they understood the benefits for skin and joints. In China, these preparations were part of everyday cooking. In Japan, collagen hot pot restaurants have been popular for generations. Western consumers knew nothing about this. Then, around 2010, the Western supplement industry discovered they could hydrolyze collagen into powder, put it in a branded tub, and sell it for $40 a container. The market exploded overnight. Billions of dollars in sales. the same compound that a bowl of homemade broth provided for the cost of bones and water. Astragalus is following the exact same path. The industry does not discover new compounds. It discovers new ways to charge for old ones. The pattern repeats with every plant that heals for free. A crop that requires no annual purchase generates no revenue for seed companies. A root that releases its active compounds in boiling water generates no revenue for processing facilities. A medicine that grows in a backyard generates no revenue for the supply chain that sits between you and your health. The system does not suppress these plants through conspiracy. It suppresses them through economics. No marketing budget, no lobbyist, no television commercial, no shelf space in the pharmacy, no awareness. Meanwhile, the anti-aging industry has perfected the art of selling urgency. Every product launch comes with countdown timers, limited edition labels, and influencer testimonials filmed in bathrooms with perfect lighting. They create the feeling that aging is an emergency and their jar is the only fire extinguisher. But aging is not an emergency. It is a biological process. The root that addresses that process at the cellular level has been sitting in the same soil for thousands of years. It was not going anywhere. It is not going anywhere now. The urgency is manufactured. The root is patient. It has always been patient. 3 generations of Westerners rubbed $200 creams on their skin, while the root that actually addresses cellular aging at the telomere level sat in a language they never learned, in a medical tradition they never studied, growing wild on mountainsides they never visited. Not because anyone hid it, because no one profited from revealing it. But here is what no supplement company will ever tell you. You do not need them. Astragalus membranaceus is a hardy perennial. It grows in USDA zones 5 through 9, which covers most of the continental United States and equivalent climates across Europe. It prefers well-drained, sandy, or rocky soil. Heavy, wet clay will kill it, but poor ground is where it thrives. Full sun, moderate water during establishment. After the first year, it largely takes care of itself. And because it is a legume, it fixes nitrogen from the air into the soil. It does not deplete your garden, it enriches it. The seeds have a hard outer coat. Before planting, soak them overnight in warm water or lightly scuff them with fine sandpaper. This is called scarification, and it helps water penetrate the seed coat to trigger germination. Sow seeds directly into the ground after your last frost date, or start them indoors 6 to 8 weeks earlier in deep pots. Because the taproot grows downward fast. Space plants 12 to 18 inches apart and leave them alone. Astragalus does not want to be fussed over. It wants to be forgotten in dry ground and left to grow. Here is the part that requires patience. The root reaches medicinal potency at 3 to 4 years of growth. Traditional Chinese harvesters often wait 4 to 7 years before digging a root for maximum compound concentration. This is not a weekend project. This is a long-term investment in your health and your soil, but consider what you get in return. A single established plant produces enough root material for a full year of personal use. Harvest in fall after the above-ground growth dies back. Slice the roots thin. Dry them in open air or a low-temperature dehydrator. Store them in a jar. They keep for years without refrigeration. Brew the dried slices as tea. Simmer them into bone broth. Add them to soups the way Korean families have done for generations. The preparations are the same ones that have been used for 2,000 years because they work. No capsule machine, no subscription, no auto ship program, no branded jar with a $200 price tag, just a root, hot water, and time. If you want to expand your supply faster, let a few plants go to seed each fall. Astragalus produces seed pods that dry on the stem. Collect them before they split open and scatter. Each plant can produce dozens of viable seeds. Share them with neighbors. Start new patches in different corners of your property. Within 5 years, a single seed packet turns into a permanent rotating harvest. where you dig mature roots from one section while younger plants develop in another. The system sustains itself. No annual seed purchase, no catalog order, no dependency on any company for what your own ground can provide. And think about what a patch of astragalus in your backyard actually represents. Dried roots that store for years without electricity, a perennial crown that returns every spring without replanting, a self-renewing supply of one of the most validated adaptogenic and telomere-supporting compounds ever studied. If supply chains fail, your patch still grows. If supplement companies triple their prices, your roots still dry on the rack. If the pharmacy closes, your tea still brews. This is what real resilience looks like. Not a subscription box, not a bottle shipped from a warehouse across the country. A plant in the ground that comes back every year and gives you what the industry charges $200 to sell. Asia did not hide this root. It was never hidden. Korean grandmothers brew it into soup every winter and serve it to their families without a second thought. Japanese pharmacies stock it on open shelves next to bandages and cold medicine. Chinese herb markets sell it by the kilogram to anyone who walks through the door. Mongolian herders still carry it in their pockets the way their great-grandparents did. The knowledge was always there, always public, always available to anyone willing to look. The question was never why did Asia keep a secret? The question is why Western systems failed to notice for so long. And the answer is not conspiracy. The answer is structure. What cannot be patented cannot be sold at premium scale. What cannot be sold at premium scale cannot be marketed. What cannot be marketed does not enter public consciousness. Economic systems promote what benefits economic systems. A free root growing wild in the mountains benefits no system except the one that matters most, yours. The root does not care about market structures. It does not care about patent law or supplement branding or quarterly earnings reports. It cares about growing. It has grown in Asian soil for longer than most modern nations have existed. It survived dynastic collapses. It survived wars. It survived industrialization. It survived being completely ignored by the most expensive healthcare system in human history, and it is still there, still growing, still producing the compound that activates the enzyme that rebuilds the part of your cells that determines how fast you age. A $2 seed packet, a patch of dry soil in a sunny corner, 4 years of patience, and a root that your grandchildren will harvest from the same ground long after you planted it. That is not a supplement. That is a legacy. Here's what most people misunderstand about stem cells. They are not science fiction. They are not experimental. They are not something that only exists in a laboratory. Your body is producing stem cells right now as you listen to this. They are rebuilding your gut lining. They are repairing damaged tissue. They are replacing worn-out blood cells and generating new immune cells every single day. This is not future medicine. This is how your body has always worked. The problem is time. By the time you reach 50 years old, your stem cell production has dropped dramatically compared to when you were 20. The repair crews are still there. There are just fewer of them. They work slower. They respond less efficiently to damage. This is why wounds heal slower as you age, why your gut becomes more sensitive, why your brain feels foggier. The stem cells have not disappeared. They have gone quiet. Recognizing this decline, a lucrative private industry emerged around regenerative medicine. Private clinics have opened globally, with many patients traveling overseas to countries like Panama or Thailand, offering costly injections of concentrated stem cells derived from bone marrow, adipose tissue, or umbilical tissues. The treatment works for some, it fails for others, and it requires repeating every few months. But there is a question that almost nobody in Western medicine thought to ask. What if certain foods, prepared in a very specific way, could signal your own body to wake those dormant stem cells back up? What if the preparation method mattered more than the plant itself? What if fermentation, the oldest food technology in human civilization, was not just preserving nutrients but transforming them into compounds that trigger cellular regeneration? 5 plants, 5 fermentation methods, all of them consumed daily by hundreds of millions of people across East Asia. and every single one of them activates a different stem cell pathway. The first one has been used to rebuild a mother's body after childbirth for over 1,000 years. In the West, landscapers spray it with poison. In Korea, childbirth is followed by a tradition that has no equivalent in Western medicine. For 21 days after delivery, the new mother is cared for in a practice called sanhujori. Her family prepares specific foods designed to help her body rebuild. One of the most important is a soft green rice cake made from a plant that grows wild across the Korean peninsula. They call it suk. The Japanese call it yomogi. Botanists call it Artemisia princeps. In most of the English-speaking world, it is called mugwort. And in most American states, it is classified as an invasive weed. Korean grandmothers did not call it medicine. They called it food for recovery. They steamed mugwort into rice cakes called suk taeok. They brewed it into fermented teas. They boiled it into baths called suk tang for the new mother to soak in. The entire tradition was built around one observation that had been passed down for centuries. Women who ate fermented mugwort after giving birth recovered faster. Their tissue healed more quickly. Their energy returned sooner. Their bodies rebuilt themselves. Western medicine dismissed this as folk practice for generations. Then researchers began studying a compound called eupatylin that is found almost exclusively in Artemisia species. Eupatylin protects the stem cell niche. The niche is the microenvironment where your stem cells live, divide, and produce new cells. When that environment is damaged by inflammation or oxidative stress, the stem cells stop working. They go dormant. Eupatylin shields that environment. It keeps the factory floor clean so the workers can do their jobs. Here is the part that changes everything. Raw mugwort contains eupatylin, but fermented mugwort delivers it in a form the body can actually absorb. The rice cakes, the aged teas, the vinegar infusions, all of these traditional preparations break down the plant's cell walls and pre-digest the polyphenols before they ever enter your stomach. The Korean grandmother did not just feed mugwort to the new mother, she fermented it first. That single step is what made the difference between eating a leaf and activating a regenerative pathway. Mugwort grows across North America, Europe, and all of Asia. It thrives in disturbed soil, along roadsides, in abandoned lots, in the margins of farmland. In Korea, it is national heritage. In America, it is sprayed with herbicides, same plant, same compounds, different understanding. But mugwort targets one type of stem cell in one system of the body. The next plant goes deeper. It feeds the system that feeds every stem cell you have, and it has been hiding underground in Japanese gardens for over 1,000 years. Walk into any neighborhood izakaya in Tokyo or Osaka on a weekday evening. Before your main dish arrives, a small plate will appear on the table. Thin strips of a pale root vegetable, braised in soy and mirin, sometimes sprinkled with sesame seeds. This is kinpira gobo. It is one of the most common side dishes in Japan. Korean families prepare the same root in a dish called ueong jorim, braised in a sweet soy reduction. Hundreds of millions of people eat this root every week. Nobody calls it a health food. It is just dinner. The root is burdock. In the West, it is the plant whose burrs stick to your socks when you walk through an overgrown field. That is the only thing most Westerners know about it. The burrs inspired the invention of Velcro in 1941 when a Swiss engineer named George de Mestral pulled them off his dog's fur and examined them under a microscope. He saw hooks, he built an industry, he never once looked at the root. Burdock root contains a compound called arctigenin. In preliminary laboratory models and animal studies, arctigenin has been observed to influence neural progenitor cell differentiation, pointing to a potential mechanism for supporting cellular health in nervous tissue. These are not foreign cells being injected, these are your own cells being told to do what they were designed to do. But that is only the first pathway. Burdock root contains up to 45% inulin by dry weight. Inulin is a prebiotic fiber that your body cannot digest. Instead, it passes through your stomach and into your large intestine, where it feeds specific populations of beneficial bacteria. Those bacteria produce a short-chain fatty acid called butyrate, and butyrate has been shown to directly stimulate intestinal stem cell renewal. Your gut lining replaces itself every 3 to 5 days. That replacement depends on intestinal stem cells. Butyrate tells those stem cells to keep dividing. One route, two completely different regenerative pathways, brain and gut simultaneously. And the traditional preparation methods unlock both. Japanese pickled burdock involves lactic acid fermentation. Korean braised burdock involves prolonged cooking in acidic soy-based broths. Both processes extract arctogenin from the dense root fiber far more efficiently than eating it raw. The fermentation and slow cooking breaks down the woody cell structure and liberates the compounds that would otherwise pass through your body untouched. In the United States, burdock is classified as a weed. In the United Kingdom, it is classified as a weed. Across most of Europe, farmers pull it from their fields and discard it. The root that activates neural stem cells and rebuilds the gut lining is torn from the earth and thrown away. Burdock works from the inside out. It rebuilds the gut and the brain simultaneously. But the next plant works on a completely different level. It does not just protect stem cells, it clears the wreckage that blocks them from doing their job. And it has been sacred across Asia for 3,000 years. In Buddhist temples across China, Japan, and Korea, the monks eat a specific cuisine that has remained virtually unchanged for centuries: temple food. No meat, no garlic, no onion. Simple preparations designed to sustain the body without disturbing the mind. One ingredient appears across nearly every temple kitchen on the continent: lotus root. In China, families prepare fermented sweet osmanthus lotus root during the Lunar New Year. The sliced root is stuffed with glutinous rice simmered in osmanthus syrup and left to ferment slightly in its own sugars. In Japan, cooks prepare renkon in vinegar-based preparations that preserve the root through mild acetic acid fermentation. In Korea, yongun is braised in soy and sesame, then stored in crocks where slow lactic acid cultures develop over days. The lotus itself is the most remarkable regenerative organism in the plant kingdom. In 1995, researchers successfully germinated a lotus seed recovered from a dry lake bed in northeastern China. The seed was 1,300 years old, 13 centuries of dormancy, and it grew. No other plant species on Earth has demonstrated seed viability anywhere close to this. The plant that teaches your body to regenerate is itself the greatest regenerator in the botanical world. Lotus embryos and roots contain an alkaloid called nephrine, which has been studied in laboratory cell models for its ability to stimulate autophagy, the body's cellular cleanup process. Autophagy is your body's cellular housekeeping system. It identifies damaged cells, cells that have stopped functioning properly but refuse to die, and it clears them out. These are sometimes called senescent cells or zombie cells. They clog the stem cell niche. They take up space. They release inflammatory signals that tell nearby stem cells to stay dormant. Nephrine helps clear them, but nephrine does not work alone. Lotus root also contains unique polysaccharides that have been shown to modulate hematopoietic stem cells. These are the stem cells in your bone marrow that produce every blood cell and every immune cell in your body. Lotus root both clears the path and activates the builders. It removes the obstacles and turns on the construction crew at the same time. Fermented lotus root preparations break down the dense starch matrix of the raw root and release nephrine and polysaccharides, informs the digestive system can absorb efficiently. Raw lotus root is a nutritious vegetable. Fermented lotus root is something closer to medicine. Lotus root is virtually unknown in Western grocery stores outside of Asian markets. No supplement company has isolated nephrine into capsules. No pharmaceutical company has patented an autophagy drug derived from lotus. The compound exists in a plant that cannot be commercially controlled. Lotus grows in any pond, any lake, any container of still water with mud at the bottom. It grows wild across most of the temperate and tropical world. It costs almost nothing, and it cannot be owned. 3 plants, 3 completely different stem cell pathways: gut stem cells, neural stem cells, blood-forming stem cells. The pattern is becoming impossible to ignore. But plant number 4 is where it all started. This is the one that 500 million people eat every single week, and it contains a combination of compounds that no other plant on Earth produces in the same concentrations. Walk into any Korean barbecue restaurant tonight, watch the table next to you. Diners will pick up large green leaves with serrated edges and purple undersides. They will place a piece of grilled meat on the leaf, add a dab of fermented soybean paste, and fold it into a wrap. This is not lettuce, this is not spinach, this is perilla. Koreans call it ki-kaenip. Japanese call it shiso. Chinese call it zisu. 500 million people across East Asia eat it every single week. Most Westerners have never heard the word. In Japan, every piece of sashimi arrives with a perilla leaf underneath it. Tourists assume it is garnish. It is not. Japanese chefs place that leaf there for a reason that predates modern microbiology by centuries. The leaf kills bacteria. The leaf kills parasites. The leaf protects the person eating raw fish. Perilla contains a flavonoid called luteolin. Research has shown that luteolin promotes neural stem cell proliferation in the hippocampus, the region of the brain responsible for memory formation and learning. It tells dormant stem cells in the brain to begin dividing again. Perilla also contains more rosmarinic acid than any other culinary herb ever tested, more than rosemary, despite sharing the name of the compound. Rosmarinic acid reduces the chronic low-grade inflammation that degrades the stem cell niche over time. When the niche is inflamed, stem cells go silent. Perilla reduces the inflammation. The stem cells wake up. Korean families have fermented perilla leaves for centuries. The preparation is called kan-neep jengaji. Fresh leaves are layered with soy sauce, garlic, chili, and sometimes anchovy stock, then left to ferment for weeks in sealed containers. This is not just preservation. The fermentation breaks down cell walls, liberates bound polyphenols, and creates entirely new bioactive compounds that do not exist in the raw leaf. The fermented leaf is biochemically different from the fresh leaf. It is more potent, it is more absorbable, it is more effective. Perilla seed oil contains 64% alpha-linolenic acid. This is the highest omega-3 concentration ever measured in any plant species, higher than flaxseed, higher than chia, and omega-3 fatty acids are structural components of stem cell membranes. Without adequate omega-3, stem cells cannot divide efficiently. They lack the building material for new cell walls. Perilla provides both the signal to activate stem cells and the raw material they need to replicate, the signal and the supply in one plant. The omega-3 supplement industry generates $4 billion annually selling fish oil capsules. Perilla seed oil can be cold pressed with a hand tool that costs less than $100. No factory, no patents, no cold chain shipping. A crop that self-seeds and returns every spring. An oil that outperforms every commercial product on the market. And almost no one in the Western world knows it exists. 4 plants, 4 fermentation traditions, 4 stem cell pathways. But here is where everything converges. The 5th entry on this list is not a plant. It is what happens when you ferment a plant in a very specific way. And when you understand what it does, you will understand why fermentation was the missing key the entire time. Every summer across Japan, millions of families perform the same ritual. They buy fresh plums from local markets. They buy bundles of red shiso leaves, a deep crimson variety of perilla. They layer the plums with salt and pack the red shiso leaves between them in ceramic crocks. Then they wait. For weeks, the salt draws liquid from the plums and the leaves. The red pigments bleed into the brine. A chemical transformation begins. The liquid that accumulates at the bottom of the crock is called aka shiso su, red shiso vinegar. Most families treat it as a byproduct, something to pour off or use sparingly as a condiment. But this liquid may be the most medicinally significant substance discussed in this entire video. Red shiso contains an anthocyanin called shisonin that is found in no other commonly consumed plant. During fermentation, shisonin combines with acetic acid in the vinegar environment to form compounds with dramatically enhanced bioavailability. Research on similar anthocyanin fermentation suggests absorption rates increase 3 to 5 times compared to the raw leaf. The anti-inflammatory polyphenols, the autophagy-promoting signals, the stem cell niche protectors, all of them arrive in the bloodstream at concentrations that eating the raw leaf simply cannot achieve. This is the moment the entire video has been building toward. Look at the pattern. Every plant discussed in the last 20 minutes shares one feature. Not the species, not the continent, not the culture, the preparation. Every single one was fermented. Mugwort was fermented into rice cakes and teas. Burdock was fermented in lactic acid pickles and slow braised in acidic broths. Lotus root was fermented in vinegar preparations and osmanthus syrups. Perilla was fermented in soy brine for weeks, and red shiso was fermented into vinegar itself. Fermentation does not just preserve food, it transforms plant compounds into forms that cross the gut barrier intact, survive the acid bath of digestion, enter the bloodstream, and reach the stem cell niches in your brain, your gut, your bone marrow, and your immune system. The method is the medicine. The clay crock is the delivery system. The weeks of waiting are the manufacturing process. Asia understood this before chemistry existed as a word. No supplement company can patent a fermentation process that grandmothers have performed for 1,000 years. No capsule can replicate the complex biochemistry of a 6-week lactic acid fermentation. The supplement industry sells isolated compounds stripped from their original context. Fermentation delivers complete systems, thousands of bioactive molecules working together in ratios that no laboratory has successfully duplicated. That is why this knowledge never crossed the Pacific Ocean in a product label. There was no product. There was only food, prepared with patience, passed down through families and eaten every single day. 5 plants, 5 fermentation traditions, 5 distinct stem cell pathways: gut renewal, neural regeneration, blood cell production, niche protection, autophagy clearance. Not one mechanism repeated 5 times. 5 separate mechanisms working on 5 different fronts of your body's regenerative system. Stem cell clinics charge $15,000 to $50,000 per session. Supplement companies sell omega-3 capsules for $30 a bottle. Anti-inflammatory drugs cost patients hundreds per month. Meanwhile, the most comprehensive stem cell support system ever documented has been sitting in fermentation crocks, clay jars, and ceramic pots across Korea, Japan, and China for centuries. It costs almost nothing. It requires no prescription. It cannot be patented. Asia did not hide any of this. Korean grandmothers ferment mugwort into rice cakes and feed them to new mothers. Japanese families pickle burdock root and serve it at every dinner table. Chinese monks prepare lotus root in temple kitchens and have done so for 3,000 years. 500 million people wrap grilled meat in fermented perilla leaves every week. Millions of Japanese families make red shiso vinegar every summer and have done so for generations. The knowledge was never secret. It was never locked behind closed doors. It was always public, always available, always sitting in plain sight on kitchen tables across a continent. The reason the West never noticed is not conspiracy. It is structure. Economic systems see what benefits economic systems. Research follows funding. Funding follows patents. Patents require control. Fermented mugwort cannot be controlled. Self-seeding perilla cannot be controlled. Lotus growing wild in a pond cannot be controlled. Burdock root returning every spring in an abandoned lot cannot be controlled. And red shiso vinegar fermenting in a ceramic crock in a grandmother's kitchen cannot be controlled. What cannot be owned cannot be sold. What cannot be sold cannot be marketed. What cannot be marketed does not enter consciousness, and what does not enter consciousness does not enter the research pipeline that Western medicine depends on to validate what it already knows. But you know now. You know the plants. You know the methods. You know why fermentation is the key that transforms a vegetable into something your stem cells actually respond to. The clay jars are still sitting on kitchen shelves across Asia tonight. The same recipes, the same methods, the same results. 1,000 years old and still working, still waiting, still free.
The science confirms what traditional knowledge preserved for centuries across East Asia, from the herbal pharmacies of Beijing to the fermentation kitchens of rural Korea to the temple gardens of Kyoto. The plants were never hidden. They were reclassified. Weeds, foreign herbs, ornamental curiosities. Anything but medicine.